record this computer. All right, so there was your example. And again, if you need help identifying keywords for topics you're interested in, let me know. I will advise you right now that health topics do tend to be extremely challenging papers to read. There is a ton of jargon in them. So if you're super interested in like osteology or cancer research, do just keep in mind some of those papers might be a little bit harder to get through because there's just so much medical terminology, which is okay. But keep that in mind. If those papers are really hard, that's why they're hard for everybody. Today, we're going to be talking about chemical reactions, enzymes, and we're briefly going to be talking about how this relates to metabolism. A couple of fun links for you if you are interested. And I might pull this up depending on what we kind of have time for in class the next couple weeks. But this is a really interesting software system where you can look at three-dimensional protein structure. You can check out the different folding sites and tertiary structure, all kinds of things. So if you want to, you can play with that. And then I said this is a video that, I don't know why I wrote the word video. That's a paper, not a video. Paper about the enzyme square dancing research that I'm going to talk about in class today. So if you want to read that, you're free to. Learning objectives for today, we're going to talk about sources for energy, where that comes from. I want you to be able to define metabolism. We need to know a fair bit of terminology, as always. I want you to definitely understand how enzymes function. And then hopefully we will have time to talk about some of the interesting applications and real world examples of enzymes, which are kind of interesting. So as a lovely guiding question, if you already know the answer to this, that's cool. Keep it in your brain. But we're going to answer this throughout our lecture today. Why can't humans eat grass? Maybe it seems simple, maybe not. So we'll answer this at the end of class. Get started. As we've talked about in this course, we have these properties of life. And in order to keep life working, there is a need for energy. So as I was mentioning, those properties of life, things like growth, reproduction, response to stimuli, all of those different components take energy. Now I have the definition of energy up here for you. It's a capacity to cause change. That's kind of confusing. Essentially, energy has the ability to move something against a force. Energy can also be heat. So the definition of energy is kind of challenging to pin down, but essentially capacity to cause change is what we're going to go with. All cells require energy. And I have a kind of fun fact for you here. We know that your brain in particular needs a lot of energy. Thinking and processing takes a ton of energy, which is why you get hungry if you study for a long time, or if you do a lot of reading in a short period, you actually do need more calories because your brain burns so many calories. The human brain accounts for like 2% of your body mass, but it takes about 20% of the available energy. So it's really energy intensive, which is cool. Thank you, brain. It is really efficient compared to other processing systems, though, like computer processing systems. The brain is very energy efficient. So energy is required in order for life's processes to be maintained. There's a couple of types of energy, and two that we're going to really focus on kind of at the beginning here. So kinetic energy is what we refer to as motion energy. A classic example here are like waves moving in the ocean, and that's what I have the picture of over here for you. Other examples would be like a baseball that has been hit and is in the air as it's moving. That would be kinetic energy. If a rock has been pushed off the cliff and it's actively falling, it has kinetic energy. We also have potential energy, which is our stored energy. And this relates to its relative position based on the Earth's core. So potential energy in general tends to relate to interactions with other objects, and you have more potential energy the greater distance that you are from that core. Classic examples of things that have potential energy, like a rock on the edge of a cliff, if it's really close to that edge, it has potential energy because it could fall off the cliff and then it would have kinetic energy. Another great example of potential energy is water being held back by a dam. So there's potential energy being held there. And then finally, we could talk about chemical energy, which is energy that can be released in a chemical reaction. And this is kind of a specific type of potential energy. Now, I mentioned that potential energy relates to how far something is from a core that attracts it. If we think about molecular chemistry, we know that there's positive cores that atoms have, and then electrons are attracted to them. There's essentially electrical poles that affect the chemical energy of different molecules. So energy is stored in the bonds of molecules, essentially. That's why gas powers cars, because when those bonds are broken, it releases energy that can be used. Questions here? Okay. So where does all that energy come from? I gave you a hint here. Where does it all come from? All energy in like a biological system. Where is its origin? The sun. Yes, we love the sun. Really awesome. Plants are able to capture sunlight's energy, transform it into sugar via photosynthesis, which we'll talk about next week. And organisms are able to feed on plants. This example of a squirrel eating a pine cone here is a great example of organisms being able to feed on plants. That sunlight energy being converted to sugar to other biological molecules is super important. And essentially, energy is constantly flowing and transforming from one energy source or one energy type to another. It's flowing in between an organism's surroundings and its body back and forth through the reforming and breaking of chemical bonds. So we see this constant flow of energy in biological systems. And the way that energy is going to move through these systems is based on the laws of thermodynamics, which if you've taken like physics classes, honestly, I think they mostly probably talk about that in chemistry classes as well. That might be familiar. One important thing to note, organisms, biological organisms, living things, live in open systems. So metabolic reactions that we talk about in biology are never going to be reaching equilibrium, which is actually great because if we reached equilibrium, there would be cell death. So we are in open systems, which is important to note just in terms of these kind of guiding processes. So this lovely image of a bear here is a good example of the first and second laws of thermodynamics. So you can see this really cute brown bear over here chewing on a fish. And that fish has chemical energy as food source. It has chemical energy. When the bear eats it, that energy can be transformed into kinetic energy in the form of movement. So bear eats fish and then is able to run because the chemical bonds in that fish are transformed into physical movement or kinetic energy. Now, the first law of thermodynamics is that energy cannot be created or destroyed. It can be transferred or transformed. So this is that transfer we see. We see chemical energy being transferred to kinetic energy. Thank you. That's awesome. The second law is kind of more confusing. Our second law of thermodynamics is about these energy transfers increase kind of disorder in the universe. And that is just the way things tend to work. So this process increases universal disorder because when the bear eats the fish, some of that energy is transformed into kinetic energy when it runs, but most of that energy is lost as heat. So in reality, tons and tons of energy is being lost as heat in biological organisms all the time. We also have this fish being broken down into other products, other smaller molecules like carbon dioxide and water, for example. Those small molecules are being released also into this open universal system, therefore increasing randomness and disorder or entropy in our system. So this is how the first and second laws of thermodynamics apply to biological systems. And generally, these processes that increase entropy are energetically favorable. They're going to be considered spontaneous reactions because energy is not required to be put into that system. They're just naturally going to occur that way. Questions here? Okay. Going a bit into some of our terminology here. Metabolism is essentially the use of organic molecules and energy to support the activities of life. You could say that in another way, which I have up here for you. Breaking down and forming of molecules that goes on inside cells. Metabolism occurs in living things, and it encompasses thousands of different biological reactions. Now, metabolic processes and this breaking down of molecules to release energy underscores all of the properties of life that we've discussed. So this is a super important process. And we're going to actually talk about some of the most important metabolic processes or pathways. Again, photosynthesis is one, very, very important, and cellular respiration, which is what we're going to talk about on Monday. Those are really important metabolic pathways and reactions that convert one type of molecule to a different one. And usually it's one that can be used. So they might take something that they can't use and convert it into something they can use, or vice versa. Now, there's a couple of different categories of metabolic reactions or metabolic pathways. We have anabolic and catabolic reactions. Anabolic reactions are those where molecules are being built up or formed, and then catabolic is the reverse. That's where they're being broken down. So molecules are being broken. To build something, it takes energy, right? If you were to build a tower of blocks, you'd have to use energy to do that. To break it down, theoretically, if you swung your arm, it would still take energy, but breaking things down releases energy. And this figure down here, these little green dots are essentially just representing like atoms. And then when they're all hooked together, that would represent like a molecule. Fairly simplified example there. Questions here? Okay. So going a little bit more here, again, more terminology. It's important to understand chemical reactions in order to understand how enzymes function. So all of this is lead up to why enzymes are so important. But essentially, chemical reactions are making and breaking chemical bonds that lead to changes in the composition of matter. So talked a fair bit about reactions, but there's a technical definition for you. And again, with classification, we can have chemical reactions that release energy or chemical reactions that require energy. And we refer to chemical reactions that release energy. Those tend to be spontaneous, which means they're just going to happen naturally over time. However, this spontaneity doesn't mean they occur quickly. Spontaneous just means it's energetically favorable for that to naturally occur. An example of a spontaneous reaction is like iron rusting over time. Does that happen super quickly? Not really. Happens over many, many years, but that is still a spontaneous reaction. So a lot of times we think of this word spontaneous as like quick and jump into the moment and things like that, but it basically just means it doesn't take energy. So I previously mentioned anabolic and catabolic pathways for metabolism. I want you to think here, would anabolic pathways be exergonic or endergonic? Chat with a friend about this maybe, and then decide for catabolic reactions as well. Thoughts here? Maybe we can go back to this other slide. So remember, anabolic reactions are where molecules are being built. And remember, it takes energy to build things. So what type of chemical reaction would that be? Endergonic, cool. And so catabolic pathways or catabolic reactions would be exergonic because they're releasing energy. So these classifications of chemical reactions are kind of like a bigger umbrella, and then we can fit anabolic and catabolic reactions and pathways underneath. Now, as I was mentioning, there are processes that are just naturally going to occur. However, there are barriers that prevent that from happening really quickly. Talk about that here in a sec. Before I go to the next slide, are we feeling good here? Cool. I did forget to explain these figures. I'm not going to spend a ton of time on it because honestly, they might not be the most helpful visuals. But essentially, these are just looking at free energy, which is energy that's available to do work, and then time. And this is essentially just showing how these reactions are either going to take energy or release energy. So that's what these graphs are kind of showing relative to free energy available to do work in a system. So we're going to talk about those barriers here. So some reactions will occur spontaneously, which means they will occur without any input of additional energy, but sometimes they still need a small boost to get started, or they need a push to be going at a reasonable speed. The boost in this situation is what we refer to as activation energy. And there's certain molecules called catalysts that can actually speed up the rate of chemical reactions by lowering this activation energy. So activation energy is the energy that is needed for a reaction to occur. And we can lower the amount of energy needed for a reaction to occur. That's what enzymes do. That's their whole job. They're doing that all the time. So when we look back at this kind of graph here that I was talking about, so in some examples, we might need to get over these little humps, and that is where we need a boost to kind of get over that energy hump. And that is what catalysts and enzymes do. Cool. I have a break here for you. We're going to do more Sudoku, and then we'll talk a lot more about enzymes, which is where the fun really starts. Not that physics and chemistry isn't fun, but. I'm also going to pass out a little piece of paper for later. You don't need for some people in the past also take pictures of these and then draw the letters or the numbers on the chat. Solve them. This one later. Okay, did anybody solve this? Do you want the answer? Ready? Okay, is this what you got? Okay, we're gonna keep going. So, as I mentioned before, enzymes are catalysts. So they're proteins that help facilitate chemical reactions. They act as a catalyst. So again, this bullet point is kind of a little bit of review from that last slide, but they act as a catalyst in either building or breaking. So they don't always just build or always just break. They can do either. They can support either type of chemical reaction. And cells have thousands of different enzymes. There's a ton of different enzymes that are at work all the time in order to make sure these metabolic processes work. When we talk about cellular respiration on Monday, I'm not going to make you memorize all the enzymes associated with it, but there's something like, I don't know, 15 or 20 that are really commonly talked about in just that one process. So enzymes are really, really diverse. Very, very important. As is true with all proteins, the shape of the protein is very important to its function. So each enzyme has a very specific structure, and alterations to that structure cause serious problems with its ability to function. So enzymes are proteins. It's a key piece to remember here. They're made up of amino acids comprised and folded into very specific shapes. That's what enzymes are. They're just a certain type of protein. Okay, so this is kind of a silly example, but we can think about a traffic jam as what would happen inside your cell without enzyme regulation. The metabolic pathways would become horribly congested because certain reactions that need to occur within certain time periods would simply not be possible. So the cellular processes that are always occurring and keeping living organisms running wouldn't be possible. Shout out to enzymes. They're like the roundabout. They improve the flow of traffic. Does anyone recognize this roundabout? This is Hayes Local Roundabout. Yes, our lovely peanut. Now, this analogy only works if you understand that roundabouts actually help the flow of traffic when used correctly. But so enzyme regulation is super important to proper functioning of cells. The ways that enzymes work are by combining with their reactant molecule, which is what we call a substrate. So I bolded that word here for you. And then in this figure, we can see the substrate is that little red molecule. So the enzyme, the big blobby protein thing, is going to bind with the substrate, and it's going to catalyze a specific reaction. Once that reaction has been catalyzed, the enzyme is going to release the substrate and continue on with its life unchanged. It can catalyze reactions over and over and over and over again. Now, when that enzyme and the substrate combine together, we call that the substrate complex. So right here, we can see our enzyme substrate complex, which is where our substrate has been attached or bonded to our enzyme. And typically, the substrate bonds to the enzyme via hydrogen bonding or ionic bonding. That's how it hooks on there and holds on. And as I was mentioning, this can occur many, many times. This can cycle repeatedly. Related to ideas about protein shape and function, the substrate that the enzyme acts on is very specific. So each type of enzyme catalyzes only specific substrates. So for example, the enzyme lactase can only catalyze a reaction for the lactose sugar. It can't catalyze other reactions for other sugars, just for that lactose sugar. Now, there's a name for the specific place in the protein, because remember, enzymes are proteins, the specific place that this is occurring, and we call that the active site. So that's what's being shown here. The active site is the place where the substrate binds with the enzyme, and that's where this reaction occurs. There's a whole lot of other protein going on that's not the active site, and it contributes to the shape of the protein, of course. And this active site is formed during the folding of the protein. So remember, proteins have to get folded and they go through multiple phases of protein modification, and that's where the active sites are formed. So, for example, if there was an issue in the amino acid structure, we would have an issue with the shape of our protein, and if it was supposed to be an enzyme, that enzyme wouldn't be able to catalyze the reaction with its substrate because it wouldn't fit. Now, historically, it was kind of considered that these reactions tend to look like a lock and key with that level of specificity, which is kind of interesting because there's been recent research that shows it's a little bit more dynamic. So I'll talk more about that on the next slide. But essentially, once that enzyme substrate complex exists, or the substrate has bound to the enzyme, the activation energy can be lowered. The way it does that depends on what enzyme it is. And I have a slide about the mechanisms for how enzymes can lower activation energy. But this is kind of a simplified visualization of how an enzyme might work. So this green molecule over here is our substrate. It's going to come into the active site of the enzyme, bind here. Then the enzyme is going to release the substrate into the two products. In this example, happens to be breaking down. Once this has happened, this enzyme can start this process all over again, catalyze more reactions. Now, what's really cool is that when the substrate initially binds with that enzyme, there's a slight change in shape in the components that ensure a perfect fit. So enzymes are not rigid structures. We think of them as kind of super, square and circle, but they're really not like that. They're very dynamic. Now, this figure is super cool. This is from research where they were looking at how enzymes actually move. And for a long time, decades, bio researchers and people who were looking at the chemical structure of enzymes, they thought that it was lock and key and there was no movement, but they've recently found they tend to dance. So they literally call it dancing. And sometimes they call it, in the paper I linked on today's intro slide. They call it square dancing because of the way they grab on and then swing around in different directions. So they do move a lot, and they can have different poses that the enzymes will take. Now, when an enzyme attaches to its substrate and then binds on, it's like a nice tight handshake. That's kind of what that could be compared to. And then when it kind of adjusts after that initial handshake phase, it's something called induced fit. So there's that adjustment between the substrate and the enzyme. That's induced fit, and that allows the reaction to be catalyzed really well. Questions on enzymes so far? Enzymes are out here dancing. That's really cool. When you look at this figure, by the way, it's kind of a lot going on. You'll notice, hopefully maybe, these little pointy bits, those are arrows. Those are showing the direction of movement. And if we remember back to what our proteins kind of look like in different figures, these squiggly dark purple lines, those are our proteins. And then the big arrows and the little green arrows, those are showing directionality for how this enzyme would be moving in combination with its substrate. So kind of cool. I wanted to put this figure up here, not because I think it's the best figure ever, but because it's showing the two different types of reactions that we're kind of going to be focusing on here that enzymes are going to be catalyzing. So remember, enzymes can either break bonds, as is the case here, or they can help form bonds. So they can do both. Now, I promised to tell you a little bit more specifically about how enzymes lower the activation energy. Sometimes they can just help move the substrate molecules in the perfect orientation so that the reaction can occur. So we know that molecules occasionally have different charges or different components, different shapes matter. So they can kind of just move the shape around, and that can be helpful. This one I think is super interesting. Sometimes enzymes can actually physically stretch the bonds so that they're easier to break. Sometimes they just literally mechanically stretch and bend bonds. Sometimes they might produce or provide a conducive microenvironment. This is kind of challenging to understand maybe, but the microenvironment would be like specific pH or the specific electrochemical environment, and they could provide certain charged particles or certain charged ions to make it better for that substrate. They could change the pH, other things like that. Sometimes amino acids that are in the enzyme itself. So again, enzymes are proteins. They're made up of amino acids. And there's certain amino acids that stick out in the active site. Sometimes those amino acids and their specific side chains are going to be able to participate in the reaction and then go back to the enzyme once the reaction's over. So these are all different ways that the activation energy can be lowered. And activation energy, what is that? Remind me, what is it? What is activation energy? Why would we want to lower it? Sure. Sure, yeah, the energy required for a reaction to occur. Yep, totally agree. So it's the energy required for a reaction to occur, and enzymes can lower the amount of energy needed for that reaction to occur. And these are some of the ways that they do it. So this slide, this is the title of this slide. I just had a lot of text, so that's going to be a tough thing here. The local conditions can affect the way enzymes function. And when I say local conditions, I mean like the cellular environment or the environment that it's hanging out in. So in general, the three-dimensional structures of proteins are sensitive to their environment. And we know that enzymes reach their maximal capacity or their maximal activity within a very narrow range of pH and temperature conditions. So when I say local environment, I'm mostly talking about pH and temperature. Now, proteins are very sensitive to their local environments. Again, mostly temperature, pH. And if we change the environment that an enzyme is in, that can change its three-dimensional structure, and therefore it can change its active site. So there are changes to the active site. What does that mean for the reactions that the enzyme would normally catalyze? The active site, the shape is different. What does that mean for the reaction? People shaking their heads. What does that mean? Perfect. Yeah. So if there's changes to the active site, that's exactly right. The shape, if the shape is different, that substrate is not going to fit right. Even though they move and there's dynamicism and they dance, it still has to be the right shape. So there's changes to the environment. It can affect the ability of the enzyme to catalyze reactions. And as I've been mentioning many times on this slide, pH and temperature impact enzyme activity. Generally, higher temperatures, up to a really specific limit, tend to increase enzyme activity just because the molecules tend to bump into each other more often at higher temperatures because they're moving around more. But again, that's a really limited amount of increased temperature. And each enzyme has its own optimal local conditions. So and some enzymes like it really hot, some enzymes, like it really acidic, etc. When we think about the enzymes that would be present in those thermal springs in Yellowstone, those really, really hot thermal springs, those are going to have different optimal conditions, obviously, than enzymes that are occurring in those Arctic char that are living in really cold oceans. So something to consider there. Now this figure or this cartoon, you're the substrate to my enzyme, and nothing could ever denature us. So when a protein loses its shape, it's been denatured, and that is what makes this joke so hilarious. Nothing could ever denature us. If the shape has been changed, the reaction cannot be catalyzed, and that is what's been. Questions here? Okay, I have more about denaturing, a couple of other visuals. I don't know, this one's fine. I like this one because it shows what caused the enzyme to be denatured, but this one makes it look like it's turning into gloopy slime, which is pretty biologically inaccurate. But again, heat and pH can cause this. This is another better figure. Here we have a lovely, properly folded protein in its nice three-dimensional shape. And then things like charged particles, temperature, or pH can cause this protein to turn into a jumbled mess. That is not the intent and will not be able to serve its purpose. Okay, here's your next break. I want you to tell me your favorite song right now. No gatekeeping. So if you have a great song, you have to share with someone in this class who you haven't talked to yet. And then write your song down. I said sticky note, but then I was like, I don't want sticky notes. I want something that's not going to stick together. So on the little piece of paper that I gave you. So what's your favorite song? Share it with a partner, a friend, and then give it to me before you leave class. Doesn't have to be your favorite song of your whole life ever, just right now. Make sure to share with someone who you don't usually talk to. That's a key piece. You have to make a new friend today. Tell them your favorite song. That's fine. I'll know that's not the name of the song. Oh yeah. Hey. No, you don't have to put your name. Keep the mystery. That way when I read them, I'm like, no, it's not for points. It's just because I like to find new music and see what you guys like. Ready to keep going? Everybody get to share? Cool. All right, we're gonna keep going. I've already said this. I've already put this bullet point up on here, but just a reminder, after our break, enzymes work within a specific range of pH and temperature. Outside of this optimal range, we see decreased enzymatic activity, typically. So the rate of this catalyzed reaction is going to decrease most of the time. Now, enzymes in humans tend to have an optimized pH of about 7. However, we know that there are some specific enzymes, particularly like digestive enzymes, which is what are being showcased on this figure on the slide. Their optimized pH is 1.5. And I'm curious how you think this came to be. So pH of the stomach is 1.5. I just told you that digestive enzymes like that pH. How do you think that happened? Like one of the major processes in life. How does that occur? Think back to our characteristics of life. Which characteristic of life could allow for this to occur? Adaptation and evolution. Yeah, totally. So enzymes can adapt to their specific microenvironments over time. And as I was mentioning, in other organisms, sometimes we tend to also see them. This figure is showing the rate of 2 different digestive enzymes. So pepsin is an important one in the stomach, and then trypsin is in the intestine. Now, this figure is not super duper hard to read, I hope. We can see here rate of reaction on this y-axis, and the peak is going to be then that is optimized, and then pH down here. So trypsin, what is its optimal pH according to this graph? 8. Does everybody agree? Trypsin, cool. Okay, pepsin, what's its optimal? 2. Cool. Does everybody get how we're reading this? Okay, lovely. Now, I'm going to set you up with a problem here. Science is about problem solving. I want you to solve this problem. Now, we're going to set the scene here. It's 1960, the era of Jell-O salad. Delicious to some, horrific to others. However, you're attempting to cook a yummy meal for your family, so you read a recipe in Home Garden. It's for a Jell-O pineapple salad. Yum! You run to the store, you grab a fresh pineapple and some Jell-O to make this delectable dessert. You chop up the pineapple, you mix it with the Jell-O according to the recipe, you stick it in the fridge for 12 hours, come back the next day, and what you find is just a bowl of colored juice with pineapple chunks at the bottom. Okay, here's a scene. Following week, you go back, you're like, I really want to have this delicious pineapple Jell-O salad. So you remake the recipe. This time you get canned pineapple at the store. Same deal, you follow the recipe. 12 hours later, the dessert has set. What the heck? Okay, with a partner, I want you to solve this mystery. I want you to come up with perhaps a hypothesis here. You can't use the internet. You have to use your brain only, just your brain. Solve this. I gave you a hint here, and think about it. Talk about this with someone. What are the differences in the way the salad was made? Why would that matter? Don't use the internet, just your brain. It relates to enzymes. Don't look ahead on my slides and find the answer. Just use your brain. Okay. You don't have to like Jell-O to solve the mystery. Solve it for your family. Okay. Hearing lots of good thoughts. Okay, some people I heard preservatives. What else? What do other people think? What the heck? What happened? And pineapple is denatured. Okay, what does everybody else think? Any other thoughts? Canned pineapple's less acidic. That's really interesting thought. Any other thoughts? Okay. Now, when you say canned pineapple is denatured, what do you mean? Pineapple itself becomes denatured? Enzymes in the pineapple. Okay, yeah, cool. So pineapple has this enzyme called bromelain. Has anyone ever eaten? Do you guys like pineapple? Some people. You know when you eat a lot of pineapple, how your tongue starts to burn really badly? It's because there's literally enzymes in pineapple that digest protein, and they're digesting the surface of your tongue. So, when things become canned, that canning process takes a lot of heat. What does heat do? Denatures enzymes. Cool. So we have this enzyme. As I was just saying, this enzyme digests protein. The setting agent in Jell-O is gelatin, which is an animal-derived protein. It comes from collagen specifically. Enzymes in the fresh pineapple, we're digesting the proteins in the Jell-O. This really happens. If you try to make Jell-O salad with fresh pineapple, it won't work. Those protein digesting enzymes break down the collagen in the gelatin and it won't go. However, if you use canned pineapple, as we've already talked about, the canning process heats things up to 121 degrees Celsius, about 250 degrees Fahrenheit. So it heats it up really, really hot, and that kills those enzymes. And by kills, I mean denatures those enzymes. So that's why canned pineapple solves the problem. Questions here? Cool.
Something like caramelized pineapple and sauteing it.
If you like cooked it in a pan first, depends on how hot you cooked it. I'm not entirely sure what the specific point for bromelain to become denatured is, but theoretically, yes, if you could cook it to that certain temperature, totally. Chop up your fresh pineapple, cook it. Another benefit of canned pineapple is that there's less water. You could squeeze the water out. So fresh, absolutely. But really, it's about the enzymes. That's a good question. Any other questions here? Okay, this one with enzymes is not done yet. There's more. Enzymes have to occasionally get some help from additional other components, things called cofactors and coenzymes. Now, there are inorganic helpers and organic helpers, so organic or inorganic molecules. When an inorganic molecule, like these examples I've given you up here, zinc, iron, magnesium, are helpful to an enzyme, we call those cofactors. And coenzymes are organic molecules that help enzymes. So cofactors and coenzymes are sometimes necessary for specific enzymes to be able to effectively catalyze reactions. Sometimes these cofactors and coenzymes are going to be permanently bound to the enzyme. They might be stuck on there forever. Sometimes there might be a loose binding and kind of a detachment after the reaction occurs. So it varies. And I have some interesting examples of some of these helper molecules in action. DNA polymerase is how nucleotides, or it is a molecule that assembles nucleotides. So we'll talk a lot about DNA polymerase here in a little bit, but essentially we would not have DNA without some of these helper ions. Vitamin C is super important for collagen production. So there's enzymes that form something called pre-collagen, and vitamin C is required in order to form that pre-collagen. Pre-collagen eventually is synthesized into those full collagen fibers. Something else that's cool about cofactors and coenzymes is they can actually help regulate metabolic processes, so they can regulate enzyme activity. Most of the time, we get all of these helper molecules that we need from our diet. So vitamin C-rich foods are featured up here, for example. Generally, animal tissues tend to have more of those inorganic compounds. Now, as I had mentioned earlier, cells have a lot of different enzymes. And most of the time, if there's a molecule, there's both enzymes that build it up and break it down. So I have a question. What would happen if both enzymes, the ones that build and break the same molecule, were active at the same time? It would be, yeah, totally useless. It would not be a helpful, it would be this problematic cycle, like you said, accomplishing nothing other than using up valuable energy. So cells prevent this from happening by enzymatic regulation. And there's different ways that this can occur. There's competitive inhibition And I bolded these vocab words. And allosteric regulation. So I have one more minute and a slide for these. So enzyme activity is regulated by competitive inhibition or allosteric regulation, which is important because that ensures that opposite metabolic processes are not occurring at the same time. Competitive inhibition is where an inhibitor, if you can see here, this little blue thing is the inhibitor in this figure. It's going to actively come in and take the place of the substrate so that the substrate can't bind. So it's going to come in, attach to the active site, and then the substrate would come in and be like, no space for me. So it blocks the access. Whereas allosteric regulation is where a molecule is going to bind to the outside of that enzyme and either speed up or slow down enzyme activity. So we could have allosteric activation or allosteric inhibition. I am out of time. I do have a couple more slides, like 2. So we'll get to those on Monday. We'll stop here. Pull ourselves